All right, class, welcome back. Today we're diving into a really foundational topic, something that pops up in both embryology and clinical genetics, chromosomal abnormalities. You absolutely have to know this stuff, not just for your exams, but for when you're diagnosing patients later on. So let's get right into it.
Okay, let's kick things off with a number that should really grab your attention. Get this. It's estimated that a whopping 25%, 1 in 4, of all human conceptions has a major chromosomal defect. That's huge. In fact, it's the #1 cause of spontaneous abortions, miscarriages, especially early on. It just goes to show you how delicate this whole process is and exactly why we need to understand what's going on under the hood.
So, here's how we're going to tackle this. First, we'll lay the groundwork, what a normal set of chromosomes even looks like. Then we'll get into the main way things go wrong, a process called non-disjunction, and finally, we'll connect all that theory to the real-world clinical syndromes you're going to see. Sound good?
All right, first things first, we've got to get our vocabulary straight. You can think of this as learning the basic language of genetics. If you don't nail these terms down now, everything that comes next is going to be confusing. So let's pay close attention here.
Okay, it all begins with this concept. Our somatic cells, basically every cell in your body that isn't a sperm or an egg, they have the full set of 46 chromosomes. We call that diploid, or 2N. Now, the gametes, the sperm and egg cells, they've got exactly half of that, just 23 chromosomes. That's called haploid, or N. And of course, this is essential for reproduction, right? 23 from mom, 23 from dad.
Now let's build on that. We have two more key terms, euploid and aneuploid. Eu in euploid literally means good or true. So a euploid cell has a good set of chromosomes, an exact multiple of 23. So 46 is euploid, 69 would also be euploid. But aneuploid means the number's wrong. It's not an exact multiple of 23. And this is where we see the vast majority of clinical issues pop up.
And aneuploidy itself comes in two main flavors that you've got to know. First, there's trisomy. Just think tri, like triangle, for 3. It means you have an extra chromosome, so 47 total instead of 46. 3 copies of one chromosome instead of the usual 2. The opposite of that is monosomy. Mono means 1, so you're missing a chromosome, for a total of 45. You've only got 1 copy where there should be 2.
Okay, so that's the what. Now for the how. How do these numbers get messed up in the first place? Well, the main culprit, the process responsible for most of these aneuploidies, is something called non-disjunction. Let's really break down what that means.
So in simple terms, non-disjunction is just a failure to separate. During meiosis, that's the cell division that makes sperm and eggs, chromosomes are supposed to split up evenly. Imagine you're sorting pairs of socks. Normally, one sock goes in one drawer, the other sock goes in another. With non-disjunction, both socks from a pair end up in the same drawer, leaving the other drawer empty. One gamete gets 2 copies and the other gets none.
And here's a really important distinction. This is a classic exam question, by the way. It matters when this failure to separate happens. If it's in meiosis 1, the first division, all 4 of the final gametes will be abnormal. It's a total mess. But if the mistake happens later, in meiosis 2, you actually end up with 2 normal gametes and 2 abnormal ones. So the timing of that error makes a huge difference in the outcome.
Okay, enough with the pure mechanism. Let's get to the clinical stuff. How does this actually show up in patients? We're going to connect this whole idea of non-disjunction to the most common autosomal trisomies. That is, trisomies of the non-sex chromosomes. You will absolutely see these in your career. The classic example, of course, is Down syndrome, which is trisomy 21. It's caused by an extra copy of chromosome 21, and that leads to this distinct set of clinical features. Now, here's another super high-yield point. The link to maternal age. The incidence goes way up as the mother gets older. Why? Well, remember that a female's eggs are basically paused in meiosis 1 from the time she's born. The longer those eggs sit there, the greater the chance that something goes wrong with separation when meiosis finally finishes decades later.
And it's important to note that while that meiotic non-disjunction we just talked about is the cause in about 95% of cases, it's not the only way it can happen. You can also have structural issues like something called a translocation or a condition called mosaicism, or only some of the body cells have the extra chromosome. But yeah, non-disjunction is by far the most common.
This table is perfect for a quick review. It lines up the 3 most common autosomal trisomies side by side. You've got Down syndrome, Edwards syndrome, trisomy 18, and Prenatal syndrome, trisomy 13.
And what I want you to really notice is how much more severe things get as we go from 21 to 18 to 13. The incidence drops and the prognosis gets much, much worse. It's a really dramatic difference.
All right, so non-disjunction doesn't just happen with autosomes. It can happen with the sex chromosomes X and Y too, and this leads to a whole different set of syndromes. And here's a good clinical pearl for you. Generally, these sex chromosome syndromes have a much milder presentation than the autosomal trisomies we just looked at.
So let's compare two of the big ones. On one side, you have Klinefelter syndrome, which is 47XXY. These are phenotypically male, but they have an extra X chromosome, leading to issues like sterility. On the other side is Turner syndrome, which is 45X. Now listen up, this is huge. Turner syndrome is the only monosomy that is compatible with life. The only one. And even then, something like 98% of these fetuses are spontaneously aborted. It's just incredibly rare for them to make it to term.
And you can also have females with an extra X chromosome, which we call triple X syndrome, or 47XXX. What's interesting here is that the features are often so mild that it might go completely undiagnosed. From a cellular biology standpoint, a key finding is that their cells will have two bar bodies. Remember, a bar body is just an inactivated X chromosome. Since typical females XX inactivate one X, it makes sense that XXX females would inactivate two.
Okay, so everything we've talked about so far has been about having the wrong number of chromosomes. But you can also have problems where the number is correct. You've got 46, but the structure of a chromosome is messed up. So let's shift gears and look at these structural changes.
A great example is a translocation. You can think of it like a cut and paste error in a document. A piece of one chromosome breaks off and gets stuck onto a totally different chromosome. Now, if it's a clean swap with no genetic info lost, we call it a balanced translocation, and that person is usually fine. But if genetic material gets lost or gained in the process, it's unbalanced, and that's when you get problems, like that 4% of Down syndrome cases we mentioned earlier.
Okay, now this is one of the coolest concepts in genetics, so check this out. It's called genomic imprinting. We're looking at the exact same tiny deletion on chromosome 15. If a child inherits that deletion from their father, they get Prader-Willi syndrome. But if they inherit that exact same deletion from their mother, they get Angelman syndrome, a completely different disease. It's an incredible example of how it's not just about what genes you have, but who you got them from.
Another classic example of a structural problem is Cri-du-Chat syndrome. This one's caused by a deletion on the short arm of chromosome 5. And the name is actually a huge clinical clue. It's French for cry of the cat. That's because infants with this condition have this really characteristic, high-pitched cry that sounds just like a cat, which can help you remember the diagnosis.
Okay, that was a lot of information. We've gone through numerical errors, structural errors, and all the syndromes that go with them. So let's wrap it up and pull together the most important takeaways.
So when you're studying, just think of it this way. Chromosomal abnormalities fall into two big buckets. Bucket number 1 is numerical errors. The count is wrong. This is almost always caused by non-disjunction, and it gives you things like Down syndrome, Edwards, Klinefelter, and Turner. Bucket number 2 is structural errors. The count is right, but a chromosome is physically broken. That's where you find deletions, like in Cri-du-Chat, or translocations, which we saw can also lead to Down syndrome.
And I'll leave you with this one final thought to chew on. We started this whole thing by saying that 1 in 4 conceptions has a major chromosomal error. That is shockingly common. And yet, over 99% of babies are born chromosomally normal. So what does that tell you? It tells you that our bodies have an unbelievably effective quality control system that weeds out most of these errors. And thinking about how that system works, well, that's a whole other fascinating topic.